Welcome to Protocol Navigator demo. Here you can design and document your experiment by evaluating your laboratory activities. Protocol Navigator has three components. First, the inventory, where instances of materials and instruments can be created or reused. Second, the bench, which is the heart of Protocol Navigator. In bench, you can evaluate your real-life laboratory activities. Based on your activities on the bench, the design of the experiment is automatically drawn on the canvas, which is the third component of Protocol Navigator. Now let us start an experiment by loading a pre-created inventory. As the inventory is loaded, like real laboratory, the materials are now available on the shelf. On the worktop, you have a 6-well plate, and of course, there is the time. For this experiment, at time 0, we seed some cells from a flask to all 6 wells of the plate. This automatically creates a flask icon on the canvas. After some time, chemical instance 1, which is the drug, was added to well A01, 2, and 3, and different concentration of the same drug was added to well B01 and 2. No drug was added to B03 at this time. So basically, cell samples were grouped into 3 sets. First group with drug concentration instance 1, second group with 2, and the third group no drug or control. Now after some time, a die was added to label all cell samples. Next day, you want to take some images. Extra 24 hours can be added to the timeline by clicking the Add button. Now on next day, by selecting an imaging instance or a macroscope channel, images were collected. For each well, acquired images can be linked, and with each image file, one metadata file can also be linked. We do it for all 6 wells. As you may have noticed, for each activity, the timeline on the canvas is redrawn. More time difference between 2 time points, more is the wiggle. So the timeline provides a temporal pattern of the experiment. In Protocol Navigator, you can also transfer your sample from one location to another. In cell biology, this is commonly called as harvest seed event. To do harvest seed, right-click on the origin location. This will open up a pop-up window showing the origin location and all possible destinations. During the transfer, scientists often do some intermediate steps like changing cell density. Here you can describe such steps through this expanding step descriptor. By selecting destination location, in this case a tube, complete the transfer. Such transfer can be done in any format, 1-to-1, 1-to-many, or many-to-1 location. Now let us take the tube to a centrifuge machine for spinning. After centrifugation, run it through the flow cytometer to acquire some FCS files and link those files with the tube. You can save the experiment for future reuse or to share with your colleagues. You can publish your experiment with the click of a button. The publish format details all information associated with the experiment, like cell line or sample details, microscope or flow cytometer settings. And when it comes to the materials and methods section, we follow the Nature Protocol suggested step-by-step format. For each step, what, where, when, and detailed description is automatically written. For measurement steps like image acquisition, links to the data and metadata files are also given. Like Google Map, the design or practice map of the experiment is also printed. Protocol Navigator is not only for designing or documenting. It also helps to share knowledge and experience with collaborators. When your collaborators open the Protocol Navigator file, he or she not only can see the experimental design, but by clicking to any activity icon can access detailed information. For example, by clicking this camera icon, collaborators get detailed information about the image acquisition activity, and with appropriate viewer can view the image data. Now if the collaborator thinks that the centrifugation activity needs some modification, by clicking on the icon can get the centrifugation instance and change there as required. Like here, a gradual increase of RPM and some additional steps were modified. Additional activity can be added anywhere within the experiment. Suppose the collaborator thought additional labeling was required before the flow cytometer measurement, and he has a good labeling procedure. He can load that labeling procedure to the inventory, and right away it is available on the shelf of the bench, which then can be applied on the tube. Unlike real life, in Protocol Navigator, an activity can be deleted from the map by selecting the icon and clicking the cut button. For activities where skill or tacit knowledge is involved, notes in the form of free text, URL, or multimedia can be added. Like here, the labeling process, a video was added. Protocol Navigator is freely available from protocolnavigator.org. Thank you for watching Protocol Navigator demo.